Welcome back, biology students. I thought I would walk you through your review for tomorrow's quest between a test and a quiz. So let's get started. So first of all, you were asked to compare and contrast mitosis and meiosis. So that means find similarities and differences. And a Venn diagram is a sweet way to show what you know in that department. So on the test, I'll give you a matching for this Venn diagram, and you'll select the items that fit under mitosis, those that fit under meiosis, and those that would be true of both. So for example, they are both ways for cells to divide. And then the difference is what kind of cells are dividing. So mitosis divides to make somatic cells, which are body cells. Meiosis divides to make reproductive cells, preparation for sexual reproduction. Similarities, we start with the same number of chromosomes in the original parent cell. The difference comes in how many chromosomes are in the daughter cell. So mitosis, the daughter cells remain the same number of chromosomes. So if I give you 46, you end with 46. If I gave you 50, you'd end with 50. If I gave you 4, you'd end with 4. Meiosis halves the chromosome number. So if you have 46 to start with, you'll have 23. If you had 50 to start with, you'll have 25. If you had 4, you'll have 2. Get it? So on the test, you'll have to identify how many chromosomes are going to be in a daughter cell based on whether it's formed through mitosis or it's formed through meiosis. So somatic cells for mitosis, gametes for meiosis. They both have the basic set of phases. Mitosis goes through them once and meiosis goes through them twice. So you should try this on your own. You can pause the video and see if you can sort these words into the proper location. All right, let's keep going. So the main difference between spermatogenesis and oogenesis, one, obviously we know that the spermatogenesis happens in males and the oogenesis happens in females. But one of the big differences is that all 4 spermatozoa will survive, all 4 spermatids, but only one ootid, only one ovum will survive. That polar body, remember, the secondary oocyte hogs all the cytoplasm, and then it splits unevenly again, and the ootid hogs all the cytoplasm again. So in the females, only one of the gametes survive, whereas in the males, all 4 do. So gametes we know are the sex cells. We know them as egg and sperm. Sperm are the male sex cells, and ovum are the female sex cells, also known as eggs. The polar body is the secondary oocyte. So that's the one that gets the little bit of cytoplasm versus the big amount of cytoplasm. So the polar body is not going to survive to pass on. It'll exit the cell cycle and apoptosis will occur, programmed cell death. Somatic cells, your body cells, are diploid. So if you see the word somatic cell in a test question, you automatically think about mitosis and you assume the cell to be diploid in number. 2N. N is variable depending on the species. Like our N number is 26. So our 2N number, our diploid number is, what did I say? Our N number is 23. So our diploid number, our 2N number, is 2 times 23, which would be 46. Gametes are sex cells and they are haploid, so they would only have 23 for us, the N number. Zygotes are the fertilized egg, so it's 2 sets of N put together, so 2 times N, you're back to a diploid number. That's the whole purpose of meiosis, is to reduce the chromosome number so that after fertilization occurs, the cell is back to the original number. So the zygote, the fertilized egg, has a diploid number. So how does this number, how is it affected by each of these processes? So we can see mitosis keeps the chromosome number the same, meiosis reduces the number in half. So here's an example of oogenesis where we took a 2N and we reduced it to an N number. And over here we had spermatogenesis. 2N was reduced to an N number. This N combined with this N and made a 2N. So it's back to the diploid number. Fertilization returns you to your original number. Random fertilization is the idea that any sperm could fertilize any egg. And what we find with meiosis in independent assortment is that every gamete contains a variety of genes. So if you start with chromosomes from mom and chromosomes from dad, the original parent cell, right? And when you divide, you might get mom and mom, you might get mom and dad, you might get mom and this dad, you might get mom and this dad. One of those was not right. But anyways, you get every combination possible, right? So who knows? Any one of these sperm could fertilize any one of these eggs. So there's lots of variations. So we have 1 out of 64 million possible combinations. Crazy. Crossing over is another form of genetic variation. So this happens during prophase 1. Remember, if we use a number 1 or 2, we're talking about meiosis. If I don't use that number, I'm talking about mitosis. So prophase 1, homologous chromosomes get tangled up. So you have 23 pairs of homologous chromosomes. Number 1, you got from mom, and you got a 1 from dad. You got 2 from mom, 2 from dad, 3 from mom, 3 from dad. Okay, so these are homologous. They carry the information for the same traits. You can see these two carry information for A, B, C, D, and E. They are not the same, however. The genetic code isn't carrying the same. So maybe this says blonde hair and this says brown hair. Down here, we can see these get tangled up and pieces break off. So now part of the original, let's say dad's chromosome, is attached to mom's chromosome. So this creates a new variation. So that is crossing over. We talked about a number of asexual reproductions. I talked more about these in the lecture than you would have read in your book. So you might want to review the lecture on this if this sounds unfamiliar to you. But we talked about budding, which is an uneven separation. You'll remember we did a matching set to practice this. The cell divides unevenly, and the smaller one continues to divide through mitosis to create a larger organism. Binary fission is the simplest form of asexual reproduction. Bi is 2, fission, splitting in half. So this is when a cell simply splits in half. A good example is a bacterium. They divide through binary fission. Sporulation. So this is going to happen in fungus. And this is when spores are produced, and those spores will drop off and they will grow into a new fungus. Vegetative propagation, think of vegetable, right? So that makes you think of plants. All plants can, maybe not all plants, but many plants can undergo vegetative propagation where they grow new plants from a root, a leaf, a stem, a nodule, all those different things. So this is an example of one coming off of a leaf, and then it will lay down new roots and become a new organism. Fragmentation, when a piece of one organism, and this is common in starfish and planaria, when one piece is broken off, that piece becomes a new organism. So I know that this starfish didn't just grow a new arm because the arm is bigger, which tells me this was the beginning piece, right? So that's fragmentation. Parthenogenesis happens in whip-tailed lizards is our common example. Some reptiles, amphibians, fish. So it's when a female is in an environment without a male for a long period of time. They will undergo kind of a double mitosis prior to meiosis. And so they're able to produce gametes that have a diploid number and therefore able to reproduce on their own. Binary fission, so here's how that works. Binary fission, here's how that works. So you can see this bacterium replicates its DNA. All of our processes are preceded by interphase and replication. So DNA genetic code is replicated. It'll attach to the cell wall. The cell splits in half, and each new cell gets a copy of the genetic code. The overall effect of mitosis and meiosis. So you get your two daughter cells with the exact same number. They are identical. You might even call them clones. When you go through mitosis, meiosis produces 4 haploid, unique, varied, every one of them is different, sex cells. So comparing asexual and sexual reproduction, of course, asexual reproduction uses only one organism, so it's going to be a little bit easier, saves energy because you don't have to go looking for a mate. Courtship, non-issue. Don't have to worry about being turned down or being hurt in the hunt for a mate. And you have the greatest chance of fitness in the individual. So greatest chance that that individual will reproduce. Unfortunately, there's low genetic variation, which means if the environment changes, the new offspring will not be varied and so not likely to survive the new environment. And so this slows evolution. Sexual reproduction produces a lot of variation. So it's facilitating, it's helping, it's aiding in adaptation, organisms being adapted for different variations, right? So if we're varied and the environment changes, some of us are going to be well suited. Some of us are going to survive and pass that trait on. Others of us aren't, and we're going to die off. But some will survive. Unfortunately, this is energy costly because you do have to go looking for a mate. And often all of the, maybe it's a dance or building a nest or having to show certain colors can be energy consuming as well. The different rituals that go along with sexual reproduction. And so usually this sacrifices the fitness of one sex to the other. So if the male maybe is going to protect the female in the nest, the male is more likely to suffer a catastrophe. With the chromosomes, you want to be able to talk about what's happening with them in each phase of meiosis. Remember, meiosis 1, you talk about homologs. Meiosis 2, you talk about sister chromatids. So if you see sister chromatids, you can narrow down to meiosis 2. If you see homologs, you narrow down to meiosis 1. So they're already in the X form because you've already copied all your genetic code. So the original cell really just had like 2 reds and 2 blues, but you've made copies of each of them. So now you have a set, like 2 pairs of red, 2 pairs of blue, right? So these are the homologs. They carry the same genetic like information for traits, different codes like blonde versus brown. One is from mom, one is from dad, and the same is here. These 2 are homologous with each other. One's from mom, one's from dad. So during prophase, not only does the nuclear membrane disappear like it did before, but the homologs will pair up. They form a group of 4, a tetrad. So the homologs come together. During meiosis 1, those homologs are orientated on the pole, or I'm sorry, on the equator, opposite poles. So when they separate, you ensure you get 1 from each pair. You get one number 1, one number 2. This is anaphase. Anaphase 1. Homologs are pulled apart. Then telophase 1, there isn't an uncoiling of the chromosomes. They'll stay coiled up. Nuclear membrane appears. And prophase 2, nuclear membrane disappears. New spindle fibers form. New spindle fibers attach to each of those pair. Notice I've already reduced my chromosome number in half. I had 4 in this original cell. At the end of meiosis, I have 2. Count the centromeres. 1, 2. Okay, going into meiosis 2, those chromosomes are going to line up at the equator. This time, sister chromatids will be facing opposite poles to ensure you get 1 from each copy. So when these are pulled apart in anaphase 2, sister chromatids are pulled apart. And in telophase 2, you reform your nuclear membrane. Again, I've maintained the N number. I started with a 2N number. I reduced it by the end of meiosis 1. I maintained that reduced number through meiosis 2. And you should be able to put these in order. Remember PMAT. So you do PMAT 1, PMAT 2. Recognize a picture of any phase. Key, look for the X's, right? Are they in groups of 2? So the tetrads, the group of 4 chromatids, 2 chromosomes. Okay, that's going to tell you you're in meiosis 1. If you see individuals here, we know we're in meiosis 2. So an organism might go through parthenogenesis, and this is, I kind of explained earlier, is because simply the male is not in the environment. So perhaps there's a chemical signal that the male would send off that would begin a sexual reproduction process. So without males in a species, some lizards, fish, and other amphibians, we saw that in Jurassic Park, will reproduce through parthenogenesis. So I said they go through DNA replication twice. So that allows them to start with a 4N instead of a 2N. So at the end of meiosis, when they reduce the number, they're already at a 2N. So that cell is already ready to grow into a new organism. And I think this is our last topic, the tetrad versus synapsis. So we talk about this during prophase, right? Prophase 1, homologous chromosomes come together. That's how they got tangled up. That's how they exchange pieces and parts. This is called synapsis. Synapsis is a process, means coming together. Okay, so the coming together of the two homologs to form a tetrad is synapsis. So how is it that sexual reproduction is aiding in evolution? This all has to do with creating variation. One, you have independent assortment of chromosomes. So during meiosis, when they separate, they can separate in any combination, right? There's crossing over, where we increase genetic variation by trading, swapping genetic codes between mom and dad. And then there's the random fertilization. Each of these sperm is carrying a different combination. The eggs, every egg that you have carries a different combination. So depending on which egg it's fertilized by which sperm, you get lots of different combinations. Okay, so all of these variations lead to evolution because it leads to adaptation. Some are better suited for an environment than others. Okay, so if you're able to do a couple of Venn diagrams comparing asexual and comparing meiosis, mitosis, asexual, sexual. If you're able to recognize your phases by chromosome behaviors, and you're able to match different asexual reproductions with their proper organism and description, I think you should be all set. Keep your numbers straight. Mitosis keeps the numbers the same. Meiosis cuts the numbers in half. Okay? So I hope that was helpful. I'll see you tomorrow. Bye.